Recording will start momentarily. Please wait. Confirm. All right. Good morning, everybody. It's good to see you guys. And we have 61 people here today. I want to say good morning to Jaden, Sienna, and Lily, and Dinar, and Victoria, and Lucas, and Carmela, Kenny, Zachary, Chris, Caitlin, Brandy, Aiden, Allison, Hunter, Jacob, Alex, Angela, Cody, Alexa, Ethan, Arnold, Robert, Jacob, Mateo, Kenya, Junior, Heidi, Stephen, Halo, Anna, Naima, Kenzie, Caden, Alma, Hunter, Zach, Aliyah, Max, Carly, Avril, Claudia, Mila, Sadie, Sarah, Chris, JR, Julian, Lily, Jesus, Caden, Eunice, Tyler, Hazel, Antonio, Jalen, Victoria, Jahari, Julian, Michael, Tatum, Chloe, JT, Samantha. Woo, 66 people here today so far. I'm so glad to see you guys. Here is our lesson plan for today. Let me just get this out of the way. We have a bunch of standards we're covering, a big old objective, and we'll go over the student objective in a minute because I have it on the Google form, okay? But we're going to be all over the place at different thinking levels. I want to make your brains hurt. So that means you guys need to get out of the refrigerator. Believe me, I would like to be there too right now. Stop watching YouTube videos. Stop texting your friends. And get off of Snapchat and let's get on this Google form. Now, I need everybody to go to the course content, and I'll show you how to do this in a minute. Let me enable my calendar. I didn't. Just give me a second here to do that. I forgot to pull up that for you guys so you can see. All right. Now I got that. Let me enable the camera. Desktop share. Start screen share. This one. Boom. Okay. So you guys, everybody, every single person, all 68 of you guys need to go into your class. It doesn't matter whether it's a regular class or honors. You go into your class, and you see where it says course content up here? You got to go there because today's lesson is not on the plan yet. It will be, but it's not there yet. And then you go to where it says gene expression. Here's the Unity Diversity TGA, the Lactase Persistence Chug Lab that you should have already started working on. Oh, by the way, in that lab from Friday, I know the video is 2 hours long, but you get to see Mr. Hancha poke himself with the needle, okay, and take my blood glucose levels. It was an exciting lab. It turned out great. You can break up the video, in multiple sections, but I give you all the answers, pretty much. Not all the answers. There's some thinking questions that you guys need to do. And I thought it was very successful. Sorry it was so long. But, an hour of that should have been in class on Friday, but I was gone, right? So it's only really an hour outside of class that you would be working on. But here's 1.01, 1.02, 1.03, 1.04. Lactase persistence. Explanation number one is between 1.4 and 1.05. That is the one we're going to be on right here. Lactase persistence explanation one. This is what we're doing today. So I'm spending class helping you with the TGA. So hopefully you don't have a lot of time outside of class to have to work on this. Sometimes you may, but most time it shouldn't be too bad. So you're going to click on that. And I don't have the assessment on here yet. That will be forthcoming. But you click on this link right here. And there it is right there. Okay. All right. Now, here's my copy. So while everybody is getting that ready, I want to kind of try to get caught up on the chat. 72 people here today. Woo! Emmy and Lily and Ricky and Santos and Josh and Adon and Samantha, JT, Chloe. I think I mentioned some of you guys already. I think I'm caught up. If I didn't mention you, it's because I'm old and I can't keep up. But I try to find everybody. Good job today, guys. Okay, so the video for this tab, you mean on the Google form, Angela? Oh, it's embedded in the Google form. It's in the Google form, not the recordings. It might be in the recordings. I'll have to check, but it's for sure in the Google form. Actually, I don't know if it's in the recordings, but for sure it's in the Google form itself. And I know some people have already done this activity, so I know it works. Okay, so I grade the TGA by giving you a quiz on it. And to do good on the quiz, you have to do the TGA because that's where all the answers are. And you'll notice the questions in the TGA are very, very similar to the assessment questions on the quiz. Okay, so the quiz, the assessment is like a reward for doing the TGA. Is that helping? And attending class makes a big difference with that too. Allison said, very, very similar. Hint, hint, wink, wink. Thank you, Piper. She says they've been getting better. Recently, I was able to receive pictures now. Okay. Is that talking about my stuff? Maybe not. Thank you, Junior, for sharing that link. Okay, let's take a poll now. Didar, you want to put a poll up, see how many people got the form going? If you got the form going, click the check mark. If you don't yet, click the X. Now, it's the X's I'm really interested in seeing because I need to know where we are. We're 74 people here. Woohoo! And we have to welcome Diego, Elizabeth, I think I already said Carmela earlier, Carson, Kyra, Josh. I think I got everybody else. Did I get Emmy and Lily? Ricky? I remember Santos. Okay. Karsten, yay. Glad you guys are all here. 75. Woo, maybe we'll hit our 100 today. That'd be awesome. Okay, 5 people still working on it. Okay, guys, if you are having a problem, let us know in the chat. Me or the moderators will help you out. Okay, some people have shared the link in the chat that might make it quicker for you to get to. Oh, yes. Oh my gosh. Okay, you know what? No, I can change that. I can change that right here, I think. I think I can do that. Let me do that. I don't know if it'll show up yet, but oh, I spelled camels wrong. Oh my gosh. CAMLES, right? Okay. Refresh to the R if you must. Okay, well, Mr. Hansen can't spell, and my spell check's not helping me. There we go. There you go. Yeah, that's cool stuff. That's cool stuff, Didar. All right, guys. So, everybody, let's do the lesson, okay? Remember, everybody read the student learning objective out loud, please. 1, 2, 3, go. I will be able to use real life data to show how specific mutations, such as the genes that control lactase production, can increase in a human population over time, depending on the conditions that humans faced in the past as they adapted to their changing environment. I'm proud of that. I know it's a little thick. I'm proud of it though. Okay. Make sure you guys put your emails so that you can get a copy of this so that it'll save your work as you go along. Remember, they'll always submit when you're done, even if you're not finished, hit the submit button on this form. It'll send you email and then you can open up from your email and then continue working. Make sure you pick your first initial last name here because I collect in the responses up here. This is my view. I collect what people put, and then sometimes we need that data to collect it as a group like we did in the lab. All right. Okay. And a couple of these slides you've seen before in the lab from Friday, but I'm connecting to that. We're continuing. Okay. All mammal babies need to drink either their mother's milk or a similar milk formula that we've made to make it just like mama's milk to survive. All mammals can drink milk as babies, but not as adults, except some humans, just some humans. Why? What is your favorite kind of milk to drink? Click on that, please, on your form, and put it in the chat. Hunter, me and you can have a discussion about this later. I have some opinions on that too, and some scientific data to back it up. But man, we can get into good debates on that. There's a lot of people that would disagree with us. Okay. Let's just do a simple little poll. I'll do it. Who likes milk? Who doesn't like milk? If you like milk, do a check mark. If you don't like milk, do an X. And what I mean is animal milk, dairy, animal milk, animal milk. Okay. Animal sourced milk. Who likes it? Who doesn't? Animal sourced milk. Who likes it? Who doesn't? And of course, on the Google form, you can pick the kind and put in the chat the exact kind that you like the best. Strawberry, it's a plant-based milk. If you put strawberry in your cow's milk, does that mean it's plant-based? All right. Yeah, I understand. Hey, now who drinks like one of the plant-based milks, like soy, almond, coconut, all those ones? Anybody like those? Who likes plant-based milks? I didn't used to like to taste when I first tried them, but since I've gone totally plant-based, I really, really like them now. Okay, that's interesting. Cool. All right. Let's move on. Okay, what is lactase? So hopefully you've seen this slide before, but lactase is an enzyme that breaks down milk sugar, which is called lactose. Okay, and so guys, look at this word lactase up here. See the ASE on the end? That means enzyme. Whenever you see ACE on the end of a word, it means enzyme. Lact means milk. And in this picture, this light blue thing is lactase. It's a big old protein. Its job is to break down lactose. Ose on the end of the word, OSE, always, always, always means sugar. Lactose is animal milk sugar. It's made out of two sugar rings, and they go into the enzyme. The enzyme busts them apart. It breaks the bonds by adding water in there, actually. It's called hydrolyzation. And it breaks lactose into two simpler sugars called glucose and galactose. And our bodies can use glucose and galactose in our cells to make ATP. And we breathe out carbon dioxide as a waste product of that. However, our cells, we can't receive lactose directly into those cells. This enzyme is secreted into the small intestine. That's where it does its work. Now, I want everybody on this Google form explain in your own words what lactase is and how it works. And I would love it if a few people would at least put what they write in the chat. What is lactase and how does it work? Go. It's your turn. And I want to, while you guys are writing, 76, Austin, Ethan, Adon, Kenny. Oh, because people come in and out, I guess. Stephen, Roxy. I think Roxy wasn't here earlier. Glad you're here. Okay. Okay, Halo and Piper, let's focus on the lesson today. Jacob, that is an outstanding description. Outstanding. Allison says, lactase is an enzyme that breaks down certain compounds in milk so your body can produce ATP. That's outstanding too. Yes, Sienna. Maybe if you're not talking to us, there you go. Thank you. Our body breaks it down in galactose, and we are able to get ATP. Thank you, Carmela. Very good. You should be taking a quiz right now. I'm not sure. Are you talking about the Unity and Diversity quiz? Because I know some people have taken it already. Carson, you have to have the password. Passwords in the towards the end of the. I'm always going to put a password in these TGAs, guys, because I want people to open up the TGAs before they take the quiz and work on it and ideally do the TGA. Adriana, welcome. Javier, if I haven't said already, welcome. Austin, welcome. Ethan, welcome. I think I got everybody else. Okay. All right. Good job, guys. Let's move on. Now, guys, listen. This is important. Get out of the refrigerator. Quit texting your friends. Get off of YouTube videos, Snapchat. We're about to embark on something a little bit more difficult. Okay? And it's based on something that we should have learned last semester called protein synthesis. For you to understand anything else now deeper about lactase and milk digestion, which is really teaching us about gene expression, you need to have a basic understanding at least of protein synthesis, which is one of the central concepts in all of biology. Okay? Now, to do that, I'm going to play this video. It's only a couple minutes. But at the end of the video, I expect that you will be able to organize the steps of protein synthesis. And it looks better in yours than does mine. Mine, it's abbreviating, but on yours, it shows the whole thing, okay? You should know, like, the basic steps of protein synthesis. And I have an organizational thing here. And you should be able to fill in these blanks accurately. In fact, you should memorize. If you'll just memorize this, it will take you a long ways in understanding biology and protein synthesis. And we really need to have this down before we can do anything else significantly. In fact, if you don't get this right now, guys, protein synthesis, or if you don't get it soon, it's going to bite you in the behind every time we learn something new about evolution and about most everything else in biology too. So it's super important that we understand this, okay? Now, I need to, so we can go back to several different ways. You can watch the video and work on your own right now. I need to get the link and put it in the, I will play it on here in just a second. So let me stop screen share for just a minute, okay? Okay, there it is right here. We get the link. Sorry, I'm not trying to play it right now. Okay, let me see if I can put it in here for you guys. Okay, here we go. Okay, why isn't that working? Oh, I need to play it up here. I'm sorry. Now remember, a chromosome is just wound up DNA. So all that's a bunch of DNA. And the DNA is a big long string of bases, adenine, thymine, guanine, and cytosine, okay? And it's all wound up. But when it unwinds, then it can help the cell make proteins. So, guys, a gene is just a section of DNA that has the information that tells the cell how to make a protein. Different sections of DNA make different proteins, okay? Okay, now this is called transcription. This is like when you copy homework off each other, okay? This is the DNA that's unwound. TAC, thymine, adenine, cytosine. So if that is what the DNA says, then this enzyme, which is this big gold thing, gray thing, I mean tan thing in the background, is going to attach messenger RNA bases to these. A to T, G to C, except RNA doesn't have T, it has U, your cell. It's almost the same thing. So A goes to T, U goes to A, G goes to C, C goes to G, and it just reads it and it's making a copy. And the copy is the messenger RNA because the DNA can't leave the nucleus. So the cell doesn't know how to make the protein if the information is in the nucleus. So a copy of the information has got to leave the nucleus, go out into the cell. That's transcription. That's what you're watching right now. So as you can see, like the little poem I have you answer in the Google form, the sequence of DNA bases determines the sequence of RNA bases. And, which you're going to see next, the sequence of RNA bases determines, let's see what it determines. Now, this is called splicing, and this is one way of like four main ways that the cell can control or regulate the proteins that are being made. And there's molecules in charge of this and things that about our physiology that influence this. So this is where our bodies can start to influence the expression of our DNA. Those 3 bases in a row are called codons. If the messenger RNA says AGU, then the transfer RNA that says UCA on this end will be the one that gets to connect here. And it has a certain amino acid. This other transfer RNA, CAG, can only connect where the messenger RNA says GUC, because C goes to G and A goes with U, right? And it has a different amino acid, usually, not always. I mean, it's always the same amino acid. There are multiple transfer RNAs that can connect to the same amino acid, okay? Remember that chart that I showed you guys last semester? It gives you the messenger RNA, and then you're supposed to come up with the amino acids that it codes for. So the sequence of messenger RNA bases determines the sequence of amino acids. And what do we call a chain of amino acids? A protein. So the sequence of DNA bases determines the sequence of RNA bases, and the sequence of RNA bases determines the sequence of amino acids. And this sequence of amino acids determines the shape of the protein. Now, the way this folds depends on the interactions between these amino acids. Some are positive, some are negative, some are polar, some are nonpolar. And it will twist and turn depending on where those are in the sequence. And of course, there's repeating ones. And that will give the protein a shape. And the shape of the protein determines how it functions. And you'll see that later today vividly. See the shape of the protein? It's like a key in a lock. The shape of the protein determines how it functions. Okay, so let's stop this part. I want to go to screen share. Okay. And I want to get out of here. And I want to go to here. Okay, guys. So you should now be matching the steps of protein synthesis. And it's okay if you get some stuff wrong. Just do your best. You can go back and rewatch the video and fix it later. Just do your best right now. Quiz yourself, basically. So let's see how you do. Welcome, Evelyn, Zachary, Adon J, Joe Marie, Adriano Javier, Austin. I think I got everybody else. Okay. 78. Woohoo. All right, guys. I need you guys working on these 2 questions. I need you to match the steps of protein synthesis to the correct description, and then to fill in the blanks in this poem. These words, amino acids, DNA, function, RNA, and protein, go in these different blanks. So which word is A? Which word is B, which is in 2 places? Which word is C, which is in 2 places? Which word is D, which is in 2 places? And which word is E? I want to take a quick poll. How many people are working on these 2 questions right now? Okay. Yours will look a little bit different than mine, the formatting. So let me open up mine that should look like yours. Okay, it should look like this, right? Okay, it should look like this for you guys. And then this one should look like this. Okay, and if you guys haven't watched the video, okay, I think there's a Chrome in the chat. Could somebody repost that link in the chat, please? And I'll try to post it here too. I can post it. Let me post it. I think I can post it. There it is.
Friendship.
Yeah.
I wanted to ask for, I think the second question, like the one that we were just doing, the matching the steps one.
Yeah.
Are we gonna? Is that just like just for us?
What do you mean?
Like, are we gonna discuss it, or are we just gonna like do it based on like what we think is
No,
right?
I want to go over this right now.
Should we do like a poll, like the 5-star one, and see which one people think is right, or do you just want to go over it?
How do you think we can do that? I'm not sure how to go about this, to be honest.
So since there's like this one thing that like the people or teachers usually use it to like rate how your day was, or like
Yeah.
how the class was,
Right,
we can
right,
use
right,
that
right.
since
Okay, so we'll have to do it one at a time though, right?
yeah. So five polls.
Okay, why don't you guys go ahead and take over? Do that.
Okay. So we're gonna. I just posted a poll. Pick which one you think the first step is. You can go to your Google form and it's the answer that I picked is winning so far, but I guess we'll see.
Well, I guess
Mr.
it's
Hanshi,
top to bottom, right? The top one is one, the
yeah. Okay.
2, 3, 4, 5.
Does
So
everyone
top
get
to
that? Does
bottom.
everyone understand how this works?
So messenger RNA leaves the nucleus would be number 1. tRNA connects to the messenger RNA ribosome would be number 2.
Yep.
A section of DNA gene is transcribed, making a messenger RNA strand would be number 3. So we're picking the one that we think is the first step. Is it?
Okay, Yeah. I'm gonna post the info. You're not gonna tell us if we're wrong or right. You're gonna tell us
Not
at the
yet.
end. Okay. I'm gonna close this. I'm gonna go again. So, what is our second step? I'm just gonna let every single one go to like 40%.
That's fine.
Yeah, so just so it doesn't take them too much time.
Now, guys, if for some reason you haven't watched the video or haven't done this yet, and you kind of like, have to just go with us and make sure you don't skip this, that you go back on your own and do this. Make sure that you got to get this right, okay, guys? And I'll have this on the assessment too.
So we're
Ooh, this
split
one's a little
between
more
3
confusing.
answers. Yeah. Do you guys want to agree? Okay, how do we go about this, Mr. Handshake?
Let's just keep going. Let's see if they can get the third step.
Okay.
Oh.
All right. What is
That
the
was pretty
fourth
good,
step?
actually. Okay, what step are we doing now? Fourth?
The fourth one. Yes.
That one actually, the one before was actually pretty good because a lot of people that got it wrong is the one step really close to it. So that's okay. That's better. They're thinking. That's good. Okay, so we're doing the fifth. Okay.
Okay, apparently it's 5 again.
That's okay. Let's do the fifth step now.
I think we're pretty clear on what the fifth step is.
Are people choosing pretty good on that one?
Yeah.
Yeah, Chloe, I want to go through this after we're done with the polls.
Sorry if I confused you guys. I just thought this would work out a lot better
No,
than it
this
did.
is great, Didar. This is great. This is what we need to be doing. I was trying to come up with a way to do it too before you said something, because it's not easy to, turn this into a poll neuro.
I disagree with this, but most people are picking 4.
Okay. All right. So let me just kind of review it,
Okay.
and
Go
then
ahead.
I want you guys Didar argue with me if you want. Okay. The very first thing that happens is the third one down. A section of DNA is transcribed, making a messenger RNA strand. We got to get the information off the DNA first, right? Then it's this one second. The messenger RNA leaves the nucleus because the information is going to get out of the nucleus into the cytoplasm so that protein can be made. Now, these next two steps, people confused, but the messenger RNA, the third step is the messenger RNA connects to a ribosome. And after it connects to ribosome, then the transfer RNA connects to the messenger RNA at the ribosome. So a lot of people can switch these two, which is a good sign because that means you're thinking along the right way. So these two steps are close together, right? Then, so what step are we on? 4 or 5? 1, 2,
We're at
3,
4.
4, 5. The
Wait,
fifth
no, we're at
step
5.
is
Yeah.
the
Okay.
amino acids connect, forming proteins. Because the transfer RNAs get in the ribosomal sequence, and that allows the amino acids that they're carrying to connect to each other and leave the ribosome in the chain. So, Didar, what was your discrepancy?
Like, what did I pick?
Yeah, you said you wanted to argue one.
I did. Oh, no. Okay. When they all picked 4 at the last one, I thought they were. I disagreed with that, but then I realized, oh, never mind. I think I messed up on the first and third one.
Okay, cool. All right, guys. So now, let me just do a poll really quick. How do you guys feel about what we just did, protein synthesis? Now that we've gone through everything. All right. All right. That's pretty good. And remember, if you have a meh or a frowny face, go back through in your own time, watch that video, and then go through this again. Okay. It's super important. It'll help you a ton. Don't let it go by. It's like multiplication tables in math. You keep needing it, right? You just don't skip it. Okay. All right. Good job, guys. Good job. Now, I want you guys to tell me, see, Didar, how can we do a poll for this one, Didar?
Sorry. Okay, we're gonna do the same system. So the sequence of blank. We're gonna. How will we do this? Okay, so number 1 would be amino acids, right? Okay,
Yeah.
we're gonna do that then.
2 is DNA, 3 is function,
Yep.
4 is RNA, 5 is protein. So you're looking for A, right?
No. I'm just asking how we're gonna do this.
Yeah. So 1 would
Same as last time,
be
pretty much. Yeah.
amino acids, 2 DNA, 3 function, 4 RNA, 5 protein. So letter A is which one of these?
1. We're actually pretty split on this one.
What do people pick?
Well, I mean, 1 is the leading answer, but B and C are tied, or 2 and 3 are tied. Okay, I'm gonna post it.
It's actually, I want to go ahead and say it. It's DNA, guys. The sequence of DNA bases. That's where we start off. So maybe the poll was confusing, but A is DNA.
Okay, so I'm gonna post the second. Should we keep doing this, Mr. Hanchett?
Yeah, let's keep going.
Okay, so I'm gonna post the second poll, and then we're gonna go on to bases determine the sequence of blank. Okay.
So what's B basically? Which one is B? And there's not a 5, obviously. Oh, yeah, there is. Oh, so maybe I picked the wrong one now because I counted wrong.
We'll see. So the leading answer is RNA. Nope. Let me post one more. Okay. We're a little unsure on this one. What does this pull for? Oh, okay. Oh, okay. So, are you guys all on the same slide as I am? No, you're fine. You're fine. We're at the bottom one on the second page, and we're trying to see what and the sequence. Oh, wait, no. Bases determine the sequence of C, so we're trying to find what C is. Does that make sense? So, for example, number one is amino acids, and we're just going down the list. Okay. Okay. I'm gonna post it. So we're thinking it's what? 3 for 3. Mr. Hanchett. Oh wait, never mind. Sorry. I think we're really confident on this one. Proteins. Function. Oh, sorry. I mean, we were pretty confident. We got 5 for 5 pretty much. Yeah, 5 for 5. What does Mr. Hanchett say? Oh yeah, we make him proud to be an American. So is one like a recessive trait? Okay. Mr. Hanchett, since no one's gonna speak up about it, I'm just gonna take one for the team. Are both the are is this lab and that recording towards the same assignment? Because I think we're all a little confused on that. Okay, right. At the end of the at the end of the class, would you be able to follow up on that and show us where the recording is? Okay, thank you. Okay, thank you.
The Unity and Diversity has its own quiz. Every TGA is going to have its own little quiz, okay? Own assessment. I haven't posted. I've only posted one, Unity and Diversity. The other two haven't been posted yet. Okay. Now, guys, listen. I need everybody's brains right here for a second. Okay. Stop, get out of the refrigerator. Stop watching videos. Stop texting. Look at this right here. We have the lactase enzyme that humans produce. That turns off in some, not others. But inside the bacteria, they have their own enzymes to break down lactase. The processes are a little bit different. I want to show you right now what happens inside the bacteria. And it's a little bit different. Then I want to show you what happens inside the human. I won't get through all this today, apparently. I want you to know that what I'm about to show you about what happens inside the bacteria is also in the course content. It's one of the lessons. I think it's 1.04. In fact, I'm getting the images from the online content because I wanted it to look the same to you. So I'm helping you with some of the lessons in the online content right now, okay? So the bacteria, here's the bacteria in your gut. He says, we digest milk differently than you humans. We have a lac operon system. That's what it's called. Our system is much simpler than yours. Here's the bacteria. Here's all the DNA in the bacteria. So look, in the human gut, these bacteria live, and inside the bacteria is DNA, and they have their own way of breaking down lactose. They break down lactose for themselves. And when they do that, it causes us problems. So here's what happens. If we take the DNA inside the bacteria and string it out, there's a little section of it that codes for the enzymes that break down lactose. There's 3 genes, and they each make a separate protein that does different things with lactose to break it down. Okay? But also on the DNA are 2 other sections. One section is called the promoter, and the other one's the operator. The promoter is where the enzyme that makes the RNA to read the DNA attaches itself and starts to read the DNA. The operator is a switch that either switches that on or off. This is how we can control the production of the enzymes. Step number 2. The cool thing about the bacteria is the bacteria don't make the enzymes if there's no milk, if there's no lactose. If that is true, if there's no lactose, then this protein called a repressor protein connects to the operator section. If there's no lactose, it will connect here. That's like a break. What that means is the RNA polymerase can't attach and read this DNA to make the gene. But it doesn't need to, right? Because there's no lactose anyways. So this prevents the DNA from making the enzymes break down lactose unnecessarily when there's no lactose present. Then, okay, so now we need to answer the question. And I lost the chat somehow. Okay. Let's say the top one is A, the second one's B, then C, then D. So yeah, let's answer the question, guys. I want to see if you guys are paying attention. Okay, how are we doing? And the right answer is C, the repressor molecule. Very good. Thank you, Didar. Okay, now I want to get to this one little section and we'll end today, okay? However, when there's lactose milk sugar, something different happens. These little green balls represent lactose sugar molecules. Those double ring sugars, lactose. If lactose is present in the gut, that means probably the person's not digesting it and there's plenty for the bacteria. So when it's present, the lactose molecules will connect to the repressor. Now, it's now showing you here is that when it connects, remember, this repressor is a protein. This blue thing's a protein. It has a certain shape. When these lactose molecules connect to the repressor protein, it changes the shape of the protein. It tweaks it. It changes its shape. When it does that, that makes it to where that repressor protein cannot connect to the DNA anymore. It's like a key in a lock. We've changed the shape of the key. Now it doesn't work anymore. What that does, that opens this up. So now this other protein enzyme, the RNA polymerase, which is the enzyme responsible for making RNA, now it's allowed to read the DNA and transcribe, go through transcription, which is what we learned about at the beginning of the class. Now we can make a messenger RNA so that that messenger RNA can go out to the cytoplasm of the bacteria cell and tell the bacteria cell how to make those enzymes to break down lactose. So this is how the presence or non, so watch what happens. So now the RNA polymerase reads the DNA, and this green thing is the messenger RNA that is being transcribed. So look, the sequence of DNA bases determines the sequence of messenger RNA bases, and it's going to go out to the cytoplasm of the bacteria, connect to a ribosome, and the sequence of these messenger RNA bases will determine the sequence of amino acids that make up these enzymes, and the sequence of amino acids determines the shape of those proteins, and those shape of those proteins, those enzymes determine how they function in breaking down lactose for the bacteria to make ATP. Whew. So the presence of the lactose controls whether these enzymes are being made or not. All right, here's the exit ticket. Last question. If you're still here, Dinar, make a poll, please. Which molecule connects to the repressor, then removing it, allowing RNA polymerase to transcribe messenger RNA so the cell can make lactase? So what connects to the repressor, changes its shape, and has it detached from the DNA? And this is our exit ticket for today. If you got to go, go, but hopefully answer this question, then go. You guys are doing a great job today. There's some heavy stuff, and we still got to do the most difficult part. But that's okay. We can get it done between next class and lab. All right, and the answer is B, the lactose molecule itself. Okay? Good job, guys. I want to go ahead and stop the recording and answer any questions that you guys have. You guys did a great job today. You made me proud to be an American. Treat your other teachers really well, like you do me. I hope you learned something. Don't forget to do the lab, TGA. And make sure this one, you can finish if you like, but I will also incorporate this into the next couple lessons. But it would not be a bad idea to finish this, but do the lab first. If you haven't done the Chug Lab TGA, do that one first before finishing this one. Okay?